Hi, everyone. My name is Monica Moore, and I am an infertility nurse practitioner. And today we are going to talk about PGT, pre-implantation genetic testing. And I have a whiteboard here kind of in the background, which you'll see. But I'm going to go over a couple of slides to hopefully explain this process, which has been increasingly more common in a way that's easy to understand. So let's start with the slides and some images, and go from there. Okay. So, as I said, what PGT stands for, and what that means is we have the ability to now test embryos, which are at what's called a blastocyst stage, which we'll talk about prior to implanting or transferring them back to you, so that we can make sure that they have the right number of chromosomes. There's a couple of terms that you might hear. One is called euploid. That means there are 46 chromosomes, the XX or XY, depending on if female or male. The other term is aneuploid, and that means that there could be an extra or missing or several extra or missing chromosomes. Most of the time, when blastocyst is aneuploid, it's what's called lethal. It means either implantation won't take place, a pregnancy won't be achieved, or in a few chromosome cases, a pregnancy can be achieved, but because a chromosome contains so much genetic material that the resulting child is usually profoundly affected. So the ability to be able to test to make sure that blastocyst is euploid prior to transferring back, I would say, is one of the most recent processes that really revolutionized infertility, the infertility field, and gave people a much better chance of achieving a pregnancy. So we start with an egg retrieval in order to be able to obtain blastocysts, which is the cell stage at which an embryo implants. We need to start with eggs. And we give you stimulating medications, as I'm sure many of you are aware. And we want to make as many eggs as possible that cycle. And just a caveat, we're actually not making eggs. The amount of eggs that come from your egg supply is inherent to you, and that changes per person, and it changes within the person per month. So instead of one egg becoming a dominant follicle, which would normally happen and ovulating, we keep the other several eggs, and how many of those are depends on you and your age, from being reabsorbed into the body. We salvage those, so to speak, with the stimulating medications before they have a chance to become dominant. We want a whole bunch of dominant follicles so that we can go in and retrieve them. So the retrieval is basically a transvaginal ultrasound with a needle attached. It goes into the vagina, through the vaginal wall, and into the ovary. And what you can see from this picture up here is as an egg grows and matures, it starts to make the follicular fluid start to increase. And what you might see when they're measuring your follicles, because eggs are microscopic in the morning, is a diameter of this, what we call follicle, fluid-filled sac that holds an egg. As the diameter increases, we assume egg maturity. So once it reaches a certain number, for often between 18 and 20 millimeters per clinic, somewhere around there, we then stick the needle into the follicular fluid, apply light suction. The egg comes out with the follicular fluid where it's put in a petri dish for the embryologist to identify it. You will know the day of the retrieval, how many eggs are retrieved, but you don't know till the next day how many are mature and how many fertilize. So all we know the day of egg retrieval is how many eggs retrieved. Since there is a natural attrition rate, meaning the amount we start with in terms of eggs to the amount of blastocysts that we get, there's a big jump or reduction. We want to start off with as many eggs as we can, hence why you're taking the stimulating medications. So this is a picture of a mature egg. We know that, and this is the embryologist see. You have to see it under the microscope because we have an oocyte here, and we have what's called a polar body. So the presence of this polar body tells us this is mature egg. This is the earliest form of an embryo called a zygote. And you can tell because there's one pronucleus or PN here from the male, one from the female. So this is called a 2PN, or a normally fertilized embryo. And again, this is called a zygote, the earliest form of an embryo. And then what you can see as the days progress is division of these cells. So here might be a day 2 where you have 4. Day 3, embryos often have 8 to 10 cells. After day 3 into day 4, which the cell stage is called a murula, the cell division is so rapid that we go from 8 to 10 cells to the blastocyst stage, which has hundreds of cells. So this is a blastocyst, and it's what I tried to draw on the board there. And the blastocyst consists of an inner cell mass, which will become the baby, trophoblast cells, which are going to become the early placenta, and what's called a blastocele cavity. So even though you can't tell in this picture, there are hundreds of cells here. The premise of PGT testing is that this blastocyst came from basically the proliferation of cells in a single embryo. So the same cell line, the same genetic component that is in the inner cell mass of the baby should also be in the trophectoderm or early placental cells. These are the cells that we want to sample so that we don't have to harm potentially the inner cell mass, which is going to become the baby. So by sampling the trophectoderm cells, we should be able to tell the genetic makeup or chromosomes of this blastocyst. In 99.5% of the time, there is a small chance called mosaicism, which is a different talk, where this cell line does not represent, or multiple cell lines are in this blastocyst. But the majority, which is always why when you look at your PGT consents, it says, even though you do PGT, if you are offered and would take up your obstetrician on first trimester screening to really check to see that this embryo is in fact euploid, we still say that if you were going to partake in that if you didn't do PGT, you should still do that because of that very remote chance that these trophectoderm cells don't represent what's in the inner cell mass. This is a picture of an embryo biopsy. So this is a holding pipette. Again, this is under a microscope. And the embryologist will orient the blastocyst so that the inner cell mass is close to the holding pipette. And then this is a pipette that we can take about 3 to 5 trophectoderm cells out off of this blastocyst. What happens then to the blastocyst is it gets frozen and it stays at your center so that we are holding it there. And each blastocyst is very distinctly organized and named and numbered, numbered, not named. So not only do we look to see kind of how does the blastocyst look to us, we give the inner cell mass a grade, we give the trophectoderm cells a grade, but we also number it. So if you have 5 blasts, they're numbered 1 through 5. And that way, when we get the results back, these cells that are biopsied get put on a slide and sent to a genetic lab. The genetic lab will come back with embryo number 1, and I'll show you these results, is euploid 46XY. Embryo number 2 is aneuploid, etc. So then we know exactly the embryos that we have in our lab, how they correlate with their genetic component. The other interesting thing and why PGT has become so important to us is that blastocysts, I'm going to go back to a blastocyst, can look really good to us under the microscope and get really good grades. So we grade an inner cell mass in A because it looks good to us under the microscope using a certain metric. We grade trophoblast cells an A or a B. That's a really good looking blast. It does not, though, we found correlate necessarily with the fact that it's euploid. So before we had PGT, we would transfer back quote unquote good looking blastocysts. But what we found is those good looking blastocysts may be genetically abnormal. And the CC blastocyst may be the only one that's genetically normal. And so that person either isn't getting pregnant or is having pregnancy losses based on that, even though obviously that was not someone's intention. Okay, so the blastocyst remains frozen. What is a very common attrition rate is maybe we retrieve 10 eggs. 8 of those are mature. 6 of those fertilize. Those 6 will continue to grow along that cell stage progression that I showed you in a previous picture. And maybe only 4 of those get to blast. or the blastocyst stage. Those 4 then are the ones that are being biopsied and frozen. Now, we don't know what percentage of those are going to be euploid or normal. So we start off with as many as we can get so that we have as many to biopsy. And there are cases where either the embryos don't get to a blastocyst stage, or if an embryo or two does get to a blastocyst stage and we biopsy it, we find out it's chromosomally not normal and you don't get a transfer. That can be incredibly disappointing news. But what we found is that when we didn't do PGT and we did transfer these blastocysts, again, either the person wasn't pregnant or they had a pregnancy loss. This is an example of PGTA results. PGT, the A in this stands for aneuploidy. So what we're looking for with PGTA is the chromosomal complement of the embryo. And what a report looks like is this. So here are the embryos numbered. Here is how they look to us. The first letter correlates with the inner cell mass or the early baby. The second letter correlates with the trophoblast and how they look. And you can see here that there are varying levels of good lookingness. So A being the best, C being the worst here. Then they give you the result. So this person has 1 euploid, so only 1 chromosomally normal embryo. It is a female chromosome. You can decide if you want to find that out in advance by telling your provider team and say, I do or do not want to know the sex or gender of the embryo in advance. We always know it because we have to count the chromosomes, and sex chromosomes are part of that chromosomal complement. The rest of these are aneuploid. This is mosaic, which I mentioned to you before, which means that the cells, there's 2 different cell lines in this embryo. When they did those 3 to 5 cells, one maybe came back XY, one maybe 46 XY, the other one may have come back 47 XY and had a whole extra chromosome somewhere. So now we're not kind of sure about the inner cell mass component and if it's reflected in that trophectoderm cell. We do transfer sometimes low-level mosaics with genetic testing. Again, that's another conversation. Sometimes we're unable to get a result for some reason. There are cases where these can be retested, but sometimes for a lab reason or the way that the blastocyst, we're just unable to get a result using the testing methods that we have to this day. So for this particular person, they had 7 embryos make it to blast and become biopsy. That's a good number, but only 1 was chromosomally normal. So there might be a situation, and these results take approximately 2 weeks to come back from the lab. So there might be a situation where somebody has to do what some places call embryo banking and do a couple of stimulation cycles to get a couple of euploid or chromosomally normal embryos. PGTM is when we are looking to diagnose in an embryo a often, not always autosomal recessive, but a recessive or dominant, usually autosomal recessive disease in a embryo before replacing it back. For example, a couple are both carriers for cystic fibrosis. There is a 1 in 4 chance of a baby being affected by cystic fibrosis, 50% chance of being a carrier, 25% chance of being completely unaffected. And so what you can tell here is we have to get samples usually from the parents and send them off because everybody's mutation is different. Everybody is different from each other. And we still do the biopsy. We take the biopsied cells where we freeze the blastocyst, send it off to test for this particular genetic mutation of this particular disease. We get results back that are either normal, a carrier. A carrier would be transferred back if there's no normal embryos, as long as there's genetic counseling. An affected embryo, where again, we can get no result or an inconclusive result for these. You can do, if you are somebody who is a candidate for PGTM, you may have had some genetic testing done on you to see if you were a carrier for certain diseases. If you were a carrier, your partner might be tested. If your partner is also a carrier, you'd be a candidate for this potentially, should you choose to do that. We can do PGTM and PGTA at the same time so that we could know whether you have normal carrier or affected, and you could also know that the chromosomes in the embryos are normal or euploid. Because if you have somebody that is, let's say, normal, meaning is not a carrier for this disease, but aneuploid, that's a problem. But if you have, let's say, a carrier that's euploid and the only embryos you have that are euploid are carriers, that couple may choose to transfer the embryos back with just genetic counseling to know what it means to be a carrier. Again, the parents were carriers, which is why they're doing this. So if the only option for the euploid embryos is that they're a carrier, then that would be helpful to know. Okay, so I'm going to stop sharing and go to my board now. So just to show you here on the board, again, this is a blastocyst. This is the cell stage at which an embryo implants. It usually takes 5 to 6 days for an embryo to get to the blastocyst stage. These are the trophectoderm cells or trophoblast cells that are the early placenta. And this is the inner cell mass. And again, we have hundreds of cells so that by only sampling, let's say 3 to 5 cells here, we are not disrupting the inner cell mass or embryo. When we first were learning the PGT process, we would start with either an earlier cell stage, where, remember the early cell stage, there were 8 to 10 cells. So taking a cell out of an 8 to 10 cell embryo can have effects. Plus, you are taking a cell that could be part of the actual baby. We've learned to be able to grow embryos to blastocyst stage. So now not only are we not disrupting the inner cell mass, but we are able to differentiate and separate out the placental cells, plus taking 3 to 5 cells of an organism that are hundreds of cells is theoretically less disruptive to that organism. So what we do is we sample these cells, we send them off to the lab, we get a result that says 46XX. We would assume that the inner cell mass, like I said, in most cases, the very great majority of cases, is reflective of the trophoblast cells. So this would be called a euploid embryo. If there were 45 chromosomes or 47 chromosomes, 45 is called a monosomy. We're missing a chromosome, one out of the 2 chromosome pairs. Or 47, an extra chromosome. So you have an extra chromosome instead of a pair. We have 3. Then this would be called aneuploid. And that would be a blastocyst that couples usually choose not to implant back. So I hope that's helpful to you. Again, we will talk about mosaicism on another talk.